Landy's going to take us away there.
Right, so for the section on the male reproductive system, if we jump into the first structure, we've got the scrotum, which is represented here in yellow on our diagram. Now, pretty much for the structure, its main role is to actually regulate the production, the temperature for healthy sperm production. So usually what we find is that the sperm's production needs to be at 2 to 3 degrees Celsius below body temperature, and that will ensure that the sperm cells that are formed will obviously be fully formed, functional, able to swim sperm cells. Now, it is important that when you stipulate the function of the sperm cell, that you state that it is for healthy sperm production, because even if the temperature was slightly higher, closer to body temperature, there will still be sperm production that takes place, but it might not be a healthy sperm that is formed. So we do have to emphasize in a testo exam that it's for healthy sperm production. Right, if we look at our second structure on our diagram, we have the testes represented here in red, and this guy is obviously the site where the sperm cells are produced. And in addition to that, it also produces the hormone testosterone that Marichen was talking about, which is released during puberty. Now, let's say, for instance, that they give you a scenario where the question states something like, what will happen to a person that wears tight-fitting underwear? You would have to relate to, obviously, how, if you have tight-fitting underwear, the scrotum would not be able to perform its job. Testicles will be too close to the body, which will obviously elevate the temperature, and it will be too warm for the sperm to fully form and produce healthy sperm cells. So that's a typical question that we've seen in past exam papers. However, you could also have approach it from a pressure perspective and a lack of blood circulation perhaps going towards the testicles, and that will also negatively affect sperm production. Right, so let's have a look at our next part. If we make a cross section through the testicle, as we've seen here, we now have a look into one of the seminiferous tubules. As we can see in the diagram, the testes is made up of many coiled tubes. Now, if we look into one of the seminiferous tubules, we will see the site where the sperm cells are produced. Now, usually what will happen is that during puberty, testosterone will be released, and that's usually the trigger point of where the sperm cells will start to produce. Now, the wall of our seminiferous tubules, where we find our germinal epithelial cells, and they are referred to as diploid cells. And then through the process of meiosis, we see the development of sperm towards the center of the tube, going through the several stages of meiosis. So we start off with having a diploid cell called the spermatogonium, and as it moves from obviously through the steps of meiosis, as we'll see in the next slide, it then develops into a primary spermatocyte, then it's referred to as a secondary spermatocyte, eventually it forms a spermatid, and then right at the end we have our fully formed spermatozoa, or commonly known as a sperm cell. Right, so the question is, how are sperm cells actually made? Now, I've already made reference to the fact that sperm is produced through the process of meiosis, which is also a chapter that you would have already completed by this point. But if we are being very specific here, we would say that the formation of sperm cells through the process of meiosis is known as spermatogenesis. And when we are talking about the formation of an ovum or ogonium, a fully formed egg cell in females, then we refer to the process as being oogenesis. But together these two terms fall under the heading gametogenesis, which when it comes to a testo exam, it might be confusing when you have been asked what do we call the formation of gametes through the process of meiosis, then obviously it's gametogenesis, the umbrella name. But if they are being specific, the formation of a sperm cell, then we are looking at the word spermatogenesis. Right, so let's have a closer look at spermatogenesis. Now, we've already done a similar image to this in the section of meiosis, so we already know that we always start with a pre-existing cell, and that pre-existing cell is diploid in nature. So in humans, we will have that diploid number represented as 46 chromosomes. So as we go through the process of meiosis 1, we are going through reduction division, which is then taking the 2N and breaking it down into 2 cells, each containing the haploid number of chromosomes. So we go from 46 to each cell containing 23 chromosomes. And this happens under the influence of testosterone in the testicles. Okay, so this is only the first half of sperm formation. Then we go through a second round of meiosis 2, as you guys would know by now, where we form 4 fully formed haploid sperm cells. So each one of them will be a viable sperm cell containing all the requirements for life. Now, in a testo exam, obviously there's a lot of information that is linked to the formation of sperm cells, spermatogenesis, and as we've seen in the previous diagram, there are also loads of scientific terms associated with the development of each of those sperm cells. So for instance, primary spermatocyte, secondary spermatocyte, and all of that. But when they ask you in a test, describe the process of spermatogenesis, then the slide that we see on our presentation, this is what we want to write down. So we don't have to go and provide all the sperm cell names. We can just quickly run through it and refer
and
to
Yolandi,
the various...
it's important to note that this is out of the exam guideline for the grade
exactly.
12s.
Yeah. So when your teacher gives you a study examination guideline from the department, this is the exact one that you will see there. So pretty much if you've maybe learned additional information to this process, this is the fundamentals that you would need to know for the examination process. So on the previous slide, we did make reference to obviously all of the sperm cells. If we just quickly go back, to all the names that they go through as they develop. Yes, you will still need to know them. So a question could be like a multiple choice question where they ask you which one represents the correct formation of sperm cells from beginning to end, and they could put these names in different orders, for instance. But when it comes down to the actual process, then we're going to talk about how spermatogenesis occurs inside the testicles, and this is under the influence of the male hormone called testosterone. So we are taking a diploid cell, which is found on the wall of the seminiferous tubule. This undergoes the process of meiosis, and eventually we only end up with 4 haploid sperm cells. So it's pretty much as simple as that. We don't have to go into too much detail with it. Right, so when we analyze the structure of a sperm cell, yet again, you can be asked to draw and label your sperm cell. And naturally, you would have to practice this, make sure that all your sections are proportional to each other. You can't have a light bulb sperm cell. Now, I'm speaking here under my own experience, having seen how my students draw the sperm cells, and it's like this, looks a bit like a light bulb at the end. So yes, definitely practice drawing one of these. There are a few diagrams that you might be asked to draw in grade 12, and the sperm cell and the ovum will be one of those diagrams. So when we run through the sections of our sperm cell diagram, we start off with having the head section to it. Then there's a part known as the middle portion or neck. Now, very important to take note of the spelling of this part section to it. I know some of the textbooks use different labels for this particular section, which I'm not going to mention, but this is the accepted grade 12 label for the middle portion. Right, and then obviously there's a tail section to it, which is this bottom half blue part that we see in the diagram. Okay, so running through the various components, we start off with the acrosome, which is in the head of the sperm cell, and this section contains enzymes. So upon the sperm cell meeting the ovum, the acrosome will open up, the enzymes will come out, and it will digest the pathway through the ovum for the head to be inserted inside it, and that will obviously be the point of fertilization. Then we have the nucleus, which is right below it, and that will be where the haploid genetic information is housed, with all the chromosomes inside it. Right, then we get to the green section here, which represents where the mitochondria is housed. And as you will recall from grade 11 and grade 10, the mitochondria is responsible for producing ATP, energy, and that energy will then be supplied to the tail section, which will allow it to swim. So that will be its way that it will propel forwards through the female reproductive system. Okay, then if we go to the last components of it, the sperm cell also contains cytoplasm, which is severely reduced, and that will allow for sufficient movement or efficient movement. And then it's also streamlined or torpedo shaped to allow for easy movement through the female reproductive system. Right, then we move to the epididymis. Now, this structure here just nestles right next to the testicle component. As soon as the testes produce the sperm cells, the spermatids, they will move into the epididymis. And this is just a temporary storage facility for the sperm where they will mature over the next couple of weeks. And then once it's ready, it will be released during the ejaculation process. So if the sperm cells are not utilized, they can be broken down, and obviously the whole process can start all over again. Okay, so in terms of examination style questions, remember to emphasize that it's temporarily stored and that we don't just say that it's stored there, because ultimately they will be broken down. Okay, from the epididymis, we move up into the next section to it, and that is represented here in the orange, which is our vas deferens. Now, as soon as the ejaculation process occurs, we will find that the sperm will be released out of the epididymis and move through the vas deferens into our urethra and eventually be ejaculated out. So this is also a question that you could experience in the test exam, where they perhaps ask you, what would be the consequence if we had to tie the vas deferens or cut the vas deferens to an individual? Now, yet again, vasectomies is not something that is discussed within grade 12, but they can still ask questions based on it. So obviously, if someone undergoes a vasectomy, vas deferens is cut, and the sperm cells will no longer be able to release out of the male reproductive system. However, there will still be seminal fluid present, but without the actual sperm cells. So just something to keep in mind should it be a topic that is discussed with an examination paper. Okay, so let's look at the glands that assist with the formation of our semen. So we start off with our first one, which is represented here in purple. That's our seminal vesicle. Now, obviously, our sperm cells, they are very metabolically active, and they require nutrients in order to make their way, in order for that mitochondria to actually propel itself forward. So one of the nutrients that we will find will be fructose, and that will provide it with the energy needed in order for ATP to be formed in the sperm cell to swim. Then we have our prostate gland, which is represented here in the blue section, and it will release seminal fluid, which will obviously have a bit of an alkaline buffer to it. It neutralizes the acidic environment of the vagina, so the vagina is actually a very hostile place for sperm cells. And that's just an additional buffer, but it also acts as a lubricant, enhancing the sperm's motility. And then we move to our third accessory gland, and this is the corpus gland, and you can see that it's got attachment place here to the urethra, and its job is to ensure that the urethra's acidic environment is also neutralized, but it also assists with the lubrication, enhancing sperm motility. And then, as we've mentioned, the sperm cells with the seminal fluid together eventually form the semen. Okay. Right, then we have the urethra, which I'm sure that you guys will recall from grade 11. That forms a communal duct for the urine to be removed from the male body, but also an exit point for the sperm cells to move out and semen to move out. So that's very different from the female reproductive system, where there's a definite urethra opening, which is completely separate from the vaginal opening of the reproductive system. Okay, then lastly we have our penis, which is obviously the external male genitalia, and it's richly supplied with blood vessels, which during the process of copulation we would require erection to take place, and that will assist in obviously inserting it into the female's reproductive system and for the sperm to then be ejaculated out.